Hello again. Today we are going to study cell division in details. If you remember, in the beginning of this unit, we said that we have two types of reproduction, which are sexual reproduction and asexual reproduction. And we connect them with two scientific concepts, which are mitosis and meiosis. And we said that mitosis and meiosis, they are very important and they are necessary to copy genetic material. In this lesson, we are going to understand how mitosis and meiosis are necessary to copy genetic material. So, here we have the main objectives of this video. Actually, in this video, we are going to understand why it's necessary to copy genetic material. And also, we will know that copying division is called mitosis and results in cells with an identical number and type of chromosomes as their parent cells. And also, we are going to study the chromosomes behaviors during mitosis, and we are going to know where mitosis takes place in the bodies, in plants and animals in mammals group. And the last thing, we are going to understand the need for a special cell division in the formation of haploid gametes. So let's start with mitosis is copying division. As you know, that characteristics are transmitted from one generation of cells to the next. So let's focus on this picture to understand mitosis. Let's start with zygote. Actually, here we have a zygote. You know that the zygote results from the fusion of egg and sperm. Then it uses mitosis process to make copy division, which produces identical daughter cells to reach into adult phase. So we start with two gametes, which are sperm and egg. They fuse together to form a zygote. Then zygote, by using mitosis process, they will divide to form an adult phase. So an adult human has about 50 billion cells, and all those cells containing the same genetic information in the zygote. So how that's happen? You know that all cells contain two sets of chromosomes. For example, human body cells have two sets of 23 chromosomes. That means the total chromosomes are 46. So 23 of two sets of chromosomes. So the total chromosomes are 46. Now, let's understand the process, how the zygote divide to form many billion cells. Focus here in this diagram. You have to know that each chromosome, we say that we have chromosomes, each chromosome has a partner. So there is no chromosome without partner. So chromosome with its partner forms homologous pairs. Both chromosomes in an homologous pair have the same genes in the same positions. So the chromosome and its partner, they form homologous pairs. And those homologous pair have the same genes in the same position. So let's start here. We have 2 chromosomes. That means we have 1 homologous pair of chromosomes. Here, we starting with DNA replication. So here DNA is replicated or copied. So each chromosome now becomes 2 identical chromatids. So each chromosome here give 2 identical chromatids. They join at the centromere. So in the middle of chromatids, we have centromere. After that, chromosomes become attached to the spindle. And the spindle here, fibers which run from one pole to another. So again, we have 2 chromosomes. They divided and replicated to give 2 identical chromatids. So each chromosomes now become 2 identical chromatids. This chromosome 2 identical chromatids, and the second chromosome 2 identical chromatids. Now the chromosomes attached to a spindle. Here we have the chromosomes, they are attached to spindle. And spindle here, fibers which run from one pole to another pole. Those are the spindle. Now, the individual chromosomes line up at the equator. They line up at the equator, the middle of the cell. Then the centromere divides and spindle fibers shorten. So in this phase, the centromeres divide and the spindle fibers shorten. Look at the spindle fiber. Here they are shortened. So that the 2 chromatids in each chromosome separate. So we say that we have 2 chromatids. So the 2 chromatids in each chromosome will separate and move to opposite ends of the cell. So this chromatid moved to this end of the cell, and the second chromatid moved to the second end of the cell. And also for the second chromosome, the first chromatid for this end and the second chromatid for this side. So here when the centromere divides, so the chromatids will move to the both ends, to opposite ends, of the cell. After moving the chromatids to opposite ends of the cell, now the cell membrane pinches in. So look at the cell membrane here in the cell. It pinches in the middle to separate the 2 sets of cells and to separate 2 sets of chromatids into 2 cells. Okay? So again, 2 chromosomes. They replicated inside the cell to form 2 chromosomes pair up here. Then each chromosome now becomes 2 identical chromatids join in the centromere. Then the chromosomes become attached to a spindle, and the spindle here, fibers which run from one end or one pole to the other. Then the centromere here, the centromere divides and spindle fibers shorten so that the 2 chromatids in each chromosome separate. They move to the opposite of the cell, to the opposite ends of the cell. After that, the cell membrane of the cell pinches in to separate the two sets of chromatids to form two new cells. Those two new cells, they are identical to one another. Look at the two cells, they are identical to one another and also identical to parent cell. This is the parent cell and those are the daughter cells, so they are the same. And also, all of them have the same number of chromosomes. So, the daughter cells and the parent cell have the same number of chromosomes. So, please try to understand the process. Let's revise it. We have two chromosomes. Okay. So here DNA is replicated exactly. Then each chromosome now becomes two identical chromatids joined in the centromere. Then chromosomes becomes attached to a spindle. Then the centromere divides and spindle fibers shorten so that the two chromatids in each chromosome separate and they move to opposite ends of the cell. After that, the cell membrane pinches in to separate the two sets of chromatids into two cells. And that will give us two identical daughter cells. And you have to know that the daughter cells, they have the same number of chromosomes and also they are identical to parent cell. Okay? Now, in your book, page 198, we have mitosis is copying division. What do you have to know from this paragraph? You have to know that chromosome has a partner. Each chromosome has a partner to form homologous pairs. Both the chromosomes in an homologous pair have the same genes in the same positions. So, the chromosome in an homologous pair have the same genes in the same position. The last thing in this paragraph, you have to know that mitosis, when cell division in which the two cells are copied of the parent cells. So, when the daughter cells are the same or copies of the parent cell, so this process is called mitosis. Okay. Now, we are going to connect mitosis with growth. So, also this in your book, the same process. Now, let's connect mitosis with growth. For example, in animals, each tissue provides its own cells when they are needed. For example, in animal cells or in mammals, for example, in our bodies, we have different organs like liver. Liver produces liver cells and heart produces heart cells. And also we have other organs like lungs. Lungs produces lung cells and so on. So you have to know that in animals, each tissue provides its own new cells. So for example, heart produces heart cells. So heart will not produce liver cells. Heart only produce what? Heart cells. Okay. The second thing you have to know that in our bodies, we have unspecialized cells are called stem cells. Okay, so we have unspecialized cells called stem cells. When those cells divide, they produce new cells that can become specialized. So stem cells are unspecialized, but when they divide to produce another cells like nerve cells, liver cells, or blood cells, they become specialized for specific functions. Here, like nerve cells, liver cells, each cell has its own function. So here I give you two examples of mitosis in animals. The first one in heart and in liver. So liver produces liver cells, heart produces heart cells. And the second example, that animal cells, or inside our body we have stem cells. Those cells, when they divide, they will produce specialized cells. So stem cells are unspecialized. When they divide, they will produce specialized cells. Now, what about the plant? Now, in plants, cell division in the cambium, so here we have cambium. So the cell division in the cambium increases the plant girth. And plant girth here means the plant gets thicker. So here we have cambium. So cell division in the cambium increases the girth of the stem so that the plants get thicker. This is the first example in plants. So cambium division or cambium cell division increases the plant girth, and girth means the plant gets thicker. And the second example, in the meristems, at the tips and the roots, the tips of shoots and roots, the cell division here leads to an increase in length. So in meristems, here we have meristems for root and meristem for shoot. So the division of meristem leads to an increase in length. So increasing length in roots and also increasing length in shoots. So here we have finished the first type of cell division. So again, we have mitosis in plants and animals. In animals, as we said, that heart produces heart cells, liver produces liver cells. And also we have stem cells. When they divide, they will produce specialized cells. In a plant, we have cambium in a stem. So when cambium cell divides, so that will increase the size of the plant, so the plant gets thicker. And also in shoots and roots, at the tip of shoots and roots, we have something called meristems. So those meristems increase the length of the shoots or roots. Okay, now let's start with the second type, which is meiosis. You know that during sexual reproduction, two gametes fuse to form the zygote. So we have two gametes, they fuse to form a zygote. Focus in this diagram, you have to know that each cell of an organism has a fixed number of chromosomes. Okay? So, the number of chromosomes in a normal body cell is the diploid number. So in normal body cell, we have diploid number, which is 2n. Okay? So the diploid number in normal body cells. But the number of chromosomes in a gamete is the haploid number. So here we have gametes. This gamete haploid number and this gamete also haploid number. During fertilization, they will produce a zygote. Then the zygote will grow up to form a baby. So we started with 2 pairs of chromosomes in homologous pairs, and each inside the body, the normal cells, they have diploid number of cells, of chromosomes. But in gametes, we have haploid numbers, which is n here and n in the second gamete. Okay, so this picture shows reduction division ensures that the number of chromosomes in sexually reproduction organisms is kept constant. So here we have adult cells contain chromosomes in homologous pairs. Homologous pairs means diploid number, which is 2n. The female give the first haploid number, which is an egg, gamete, and the male give the second haploid number in sperms. So here we have n and n. And during fertilization, and we say that fertilization is the fusion of haploid gametes to restore the diploid number in the zygote. So during fertilization, we will produce the zygote which contains the diploid number of chromosomes. So here we have offspring, and the offspring cells contain chromosomes in homologous pairs. Homologous pairs means they have the diploid number of chromosomes. Each pair contains 1 chromosome from the mother and 1 from the father. So we have diploid. They were 2 haploids. The first haploid from the mother and the second haploid from the father. Okay? Now, let's understand the division in meiosis. In this diagram, we have the meiosis, and sometimes we call it reduction division. Actually, we call it reduction division because it halves the number of chromosomes in cells. So, reduction means decreasing the number of chromosomes. So, again, meiosis is called reduction division because it halves the number of chromosomes in cells. So, if I ask you, explain meiosis is called reduction division, this is the answer because it halves the number of chromosomes in cells. Okay, let's start the process. As we said that each parent cell contains 2 sets of chromosomes. The total number of chromosomes, the diploid number, 2n. So, a total number is the diploid number. And the diploid number, you have to know that it's double the number of haploid number. The diploid number is 2n and haploid number is 1n. Now here we have 1 pair of homologous chromosomes. 1 pair of homologous chromosomes. So we have 2 chromosomes. Actually, homologous pairs of chromosomes line up. In this stage, we have a stage here. In this stage, the homologous pairs of chromosomes line up. And that's the difference between meiosis and mitosis. Meiosis, the homologous pairs, they line up. But before, in mitosis, direct, the DNA is replicated exactly. here, before replication, they line up. So here they wrote that this is the key difference from mitosis. And you have to know that this step is very important in offering the chance of variation. So without this stage, there will be no variation. So because in mitosis, the chromosomes don't line up, so there are no variation. But here we have this step, so homologous pairs of chromosome line up, so they have chance of variation. After lining up, the DNA replicates. So here we have DNA replication. What do you mean by replication? You know that each chromosome becomes two identical chromatids. So replication, as we said before, makes a DNA copy using the DNA. So here we have DNA, we copy it, and another DNA chromosome also we copy it. Okay, so after lining up, we have DNA replication. Now, homologous pairs line up at the cell equator. Here we have the equator of the cell, so the homologous pairs line up at the cell equator. Here, spindle fibers separate whole chromosomes. So when the cell divide and the spindle shorten, spindle fibers shorten, they will separate all the chromosome. So cell divides, each cell now has only one chromosome from each homologous pair. So we have two chromosomes. Those chromosomes line up. After lining up, they replicate. After replication, the cell membrane divides. But before dividing, the spindle fibers separate the whole chromosomes. Then it separates the cell membrane to form two different cells. Each chromosome separates into two chromatids. Here we have this cell divides. Okay? Now, after the first division, here we have two cells. Each cell has a chromosome. We have another division. This division will divide the chromatids into two cells. And here actually it's not cell, it's a gamete. So here we have 4 gametes, each containing only one set. So here we have the haploid number of chromosomes. Haploid number, haploid, N, 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 N. So all of them are haploid numbers, haploid numbers of chromosomes. So the process again, 2 chromosomes, they lined up. After lining up, they replicate. After replication, the spindle fibers separate the whole chromosomes to form 2 cells. Each cell now has only one chromosome from each homologous pair. After that, each chromosome separates into 2 chromatids. So that is the cell division. And each cell has haploid number, N. And that's the difference between meiosis and mitosis. In mitosis, we produces 2 new cells have the same number of chromosomes with parent cell. But here, the parent cell is 2 chromosomes, and here the daughter cell has only one set the haploid number of chromosomes. Okay? I hope that you understand the lesson. If you have any question, you can ask me. Thank you for listening and good luck.